Hello, good afternoon. We are going to do a little recap of the key concepts that we've learned from last class. And I wasn't able to finish lecture 8 this morning with my other class, but we got close to the end. So we'll see how it goes. Quiz is next Tuesday, of course. And everything that's going to be on the quiz will be covered by the end of today's lecture. So let's take a look here. We were talking about how we can turn bacteria into little insulin factories, right? So we've located the gene, we've cloned the gene, and we talked about how we can insert the gene into the bacteria. And so let's just do a little recap here. So here we have a tube here that contains the DNA, which is the gene of interest. Right? And it has been copied many times using PCR. Okay, so we have many copies. Of course, you cannot see it with your eyes, but I'm just gonna draw it here. It's in the tube. The tube is very small. It's about this big. Okay. And what we also have would be another tube here that contains a plasmid. And we purchased the plasmid from online, from maybe like a biotech company that sell these things. Right? So the plasmid are little circles, and again, you can physically see them in the tube, which is like it would just look like water. Okay, but within that solution, you would have all these things. And what we want to do here is we want to treat the DNA with some kind of restriction enzymes. Right? And then that will allow our DNA, our gene, to have sticky ends in the end. Yeah, to create these sticky ends. So this is the gene, the insulin gene, and then these are the flanking sequence, which contains the recognition sites for the restriction enzyme, and we have created sticky ends. Now, the idea here is to treat your plasmid with the same restriction enzyme. Okay, with the same restriction enzyme. And if I do that, I am going to I'm going to open up the plasmid basically. And it's going to pop open, and we're going to have the same kind of sticky ends. Right. As long as the restriction enzymes are the same, the sticky ends that we created for the gene and the plasmid will be compatible. Right? So I'll put it here, like compatible. Right? It means like it would be able to insert itself into the plasmid. Right? Again, these have to be the same restriction enzyme. Right? Must be used to create compatible sticky ends. All right. If they're not compatible, you won't be able to insert the gene. And then all this would be for nothing. Then we will mix these things together in like another tube. We're going to add a little bit of ligase. Then what will end up happening is the gene is going to be inserted into the plasmid. Okay. So gene of interest is inserted into the plasmid. Okay. Then we're going to feed the plasmid to bacteria. Okay, you're going to have another tube of bacteria, and you're going to mix the plasmid with them. And then you can create certain conditions to encourage them to take up the plasmid. Right? But in the end, what's going to happen is here is a bunch of the bacteria. Okay, bacteria. Right? And then you're going to add the plasmid. The plasmid are these guys, right? Okay. Remember, the plasmid has your gene in it. You're going to mix them together. You're going to encourage them to take it. I'm not going to go into the details of how you can encourage them. You're just like, take it, right? And so you cannot guarantee all of them are going to take up the plasmid, but afterwards you will have some bacteria that have taken the plasmid and others would have not. Let me just copy this guy over. Right. So some would have accepted the plasmid. Some would not. When they have accepted the plasmid, we say they have been transformed. Some will not take up the plasmid. So far, that's the story. Does anybody have any question? Now, before we go any further, let's take a closer look at the plasmid. Okay, so again, you can buy these plasmids just online, right? With there are many companies that make these things. So this here in the middle, that's just the name of the plasmid. We have the origin of replication here. That's where the transcription begins. That's useful information if you're actually doing this experiment. And then we have all these things on the outside here, right? PST1 equal R1, BAMH1, alpha 1. These are recognition site for different restriction enzymes. Okay. They are like insertion site, if you would. let's pause for a second and go back. If I treat my DNA with restriction enzyme, let's say I treat it with E-core-R1, right? Then I have to treat my plasmid with E-core-R1 as well in order for them to have compatible sticky ends. So if I do that, if I actually use E-core-R1, my gene of interest is going to go right here into the plasmid. Right? If I use BAM-H, then it's going to go right here. If I use PST1, then it's going to go right here. Right? So there are multiple locations that you can insert the gene into the plasmid. Okay. So why might you choose one over the other? Well, remember, let's go back here. The restriction enzyme works by recognizing a particular sequence, right? The palindromic sequence that we learned last class. Say it's AATT. Then it only works if we actually have AATT at the end of the gene, right? And then you're going to cut the AATT, and then you're going to have the sticky ends. So that's okay. But what if it doesn't have AATT, right? What if it has, you know, something else like CCCGGG? Then E-core-R1 might not be suitable. Then I have to choose another restriction enzyme, right? Okay. So, you know, you don't always have the sequence that you want at the ends. So that's why you can't always use the same restriction enzyme. And the second reason is if the sequence, if the AATT, for example, also exists within the gene, then that's also not a good restriction enzyme to use. Because not only are you cutting the ends, you are also cutting the middle of the gene. You destroy your gene. And that's not going to work. That is why when we purchase a plasma, there are multiple options. Just in case you cannot cut your gene with E-core-R1, well, you can cut it with BAM-H. And if that doesn't work, you can cut it with one of the other ones. Okay? And if none of it works, well, then you got to buy another plasmid. Okay? We also have other stuff here. We have these antibiotic resistant genes. Those are for, we call them like selection markers. Right? It gives us a way to know, to tell the bacteria apart. How do I know which one have accepted the plasmid and which one have not? Okay, they look the same under the microscope, right? Like on a plate physically, if you look at them, they also look the same. So how do you know if they have accepted it or not? Well, in this case, AMPR and TETR gives the bacteria resistance to ampicillin and tetracycline, respectively. If the plasmid is accepted by the bacteria. Okay? When it's taken up the plasma, not only is it going to produce the gene that we want it to produce, but it is also going to produce these ampicillin-resistant genes, which makes it resistant to ampicillin, like it's not going to get killed by ampicillin, as well as the tetracycline-resistant gene. Does anybody want to ask anything before I move on? Anything you would like me to clarify? Now is a good time to ask. All right. So let's take a look at what happened. Okay. Suppose we transform bacteria with plasmid that contains a gene that was inserted using equal R1. Okay, so it goes right here. Okay. Let's take a look at what happens. Okay, so we have two groups of bacteria. Let's look at those that have not accepted the plasmid. Okay. Did not accept the plasmid. So what will happen if you try to grow them? So let me show you how you grow bacteria. Okay, we grow them in this agar play, AGAR, agar play. Right? They are, you know, the agar gives them a place to stick on, and then we pour some nutrients on them. Okay, and then let's take a look at what they look like. You know, afterwards they look like this. You know, they have they grow little colonies like that. All right. That's basically how you culture them. If you didn't know, now you do. So this is the bacterium. You know what? Let's draw a bunch of them. Why not? Okay. We have a group of resistant. They didn't want to accept the plasmid. So let's see what happens if I grow them on a petri dish. Okay, this is the dish I just showed you. Okay, and then it just have basic nutrients and nothing else. Okay. Well, if you give them basic nutrients and you let them grow, of course they are gonna grow. Okay. They're gonna grow. They're like, oh, nutrients, nice, let's grow. Okay. So lots of growth. Okay. Sometimes they call this like lawn growth. It grows so much, it covers the whole lawn. And then what will happen if you grow them in another dish? Okay, but this dish has nutrients plus ampicillin. Do you think they will grow? Or do you think they will die? What do you think? Tell me. Will they grow or will they die? Maybe I give you a poll. Okay. Oops. Let's try again. If you think they will grow, please choose A. Grow. If you think they will die, please choose B. Will they grow? Will they not grow? Okay. I have 4 response and 5 now. Anyone else before I close the poll? Okay, so we have half. Okay, 8 people now. That's not bad. 4 of you say, 5 of you say it will grow, and then 4 of you say it will not grow. Well, let's find out. So over here, it has ampicillin. Ampicillin is an antibiotic, right? Antibiotic kills bacteria. Okay, so no growth. No growth. The ampicillin will kill them all. Okay. I hope that makes sense. Ampicillin kills bacteria. Okay, these are just normal bacteria. Okay, so that is that. I'm just gonna make this smaller so I have room. Let's take a look at a second group here. Okay, second group here. Okay, these are the ones that have accepted the plasmid. They have accepted the plasmid. Okay. Transformed. They accepted the plasmid. Let's take a look at what happens if you grow them in the plate. Okay, so this would be just nutrients with no antibiotics. Do you think they're going to grow or no growth? Here, let me launch the poll again.
You said without antibiotics?
Yeah, without just normal nutrients. You think they're going to grow?
They will grow.
They will grow. They will grow like weeds, okay? Because nothing's stopping them. They're just going to grow, grow, grow. Okay, no point of doing the poll now. I just kind of gave you the answer, but you know, hopefully you agree with me that they're going to grow, right? All right. What about with the antibiotics? Nutrients plus ampicillin. This is the antibiotic. You think they're gonna grow? Let's do the poll this time. No, no growth. Okay. Anyone else? Let's just do the poll. I want to see. I want to know what you guys think. A for growth, B for no growth. Anyone else? Okay, well, let's take a closer look at this. Let's take a look at the plasmid. What's in the plasmid? Well, the plasmid has a ampicillin-resistant gene, doesn't it? Right? So it says if the plasmid is accepted by the bacteria, the gene will give them resistance to ampicillin. So if these are bacteria that have been transformed, they have accepted the plasmid, then they will gain the resistance. They will be able to grow. The antibiotic doesn't bother them. They're like, ampicillin, I don't care. I have the ampicillin resistant gene because I have accepted the plasmid. Okay, so the plasmid that they have accepted, that's why they're called transformed, right? The plasmid gives the bacteria ampicillin. Ampicillin resistance. The ampicillin is not going to stop them. Lots of growth. Is that okay? All right. So let's go back here. Remember this tube? Like we're gonna have like the bacteria, we're gonna feed them the plasmid, but like within the tube, you're gonna have a mixed population of these and these, right? They're gonna be together. I mean, if they're separate, then I don't need to select for them, right? It's because they're all together and I can't tell which one has the plasmid and which one does not, right? So when you take all these things, let me try to, just copy it over here. And then I'll copy these guys as well. One more time. So like when they're all mixed together in the same tube, okay, some have it, some don't have it. I can't tell. Okay, so you're gonna grow this in a plate, with ampicillin. Right? You will have some growth. Right? It's not gonna be like long growth. It's not gonna be a lot. So you're gonna have some growth, because only those who have accepted the plasmid will grow. Everything else will die. That's how you can select for them. That's how you can tell these ones that are growing on the plate, those are the ones we want. Those are the ones we want to keep. Those are the ones who have accepted the plasmid, therefore accepted the gene of interest. And if the gene of interest is insulin, these are the ones that will produce insulin for us. Okay? I feel like you guys have a lot of questions. Like, just hit me with them. Like, I don't know what you don't understand. So if there's something that's bothering you, just ask and then I'll clarify for you. Someone else probably have the same question in class. All right, fine. That's okay. Let's do some poll questions, and then maybe we'll find out, what is it that we do understand or we don't understand. Now I don't know if you guys have been doing those practice quizzes, okay? You definitely want to do the practice quizzes for lecture 9, to prepare for the quiz for next week. Okay, there are some good questions there. So here, a gene is inserted into the plasmid using E. coli R1. The plasmid is transformed into a tube containing E. coli. You culture all the E. coli on the plate containing tetracycline. What results do you expect? So we are inserting using E. coli R1. The gene goes right here, according to the map. That's my gene of interest. Okay. Some will accept the plasmid, some will not. So when you plate it, when you grow it on the plate with tetracycline, do you think they're going to grow or not grow? Or like some growth? What do you think? Yes, Patrice, go ahead. You were going to say something and then I interrupted you with the poll. Go ahead. If you
I was gonna answer the question.
Yes, sure. Why tell us. What do you
I
think?
think only some will grow
Yeah, and why is that?
because you just said some have the gene, not all of them.
Exactly, right? Like if you feed it to them, not all of them are gonna take up the plasmid. If they don't take up the plasmid, if they don't take up the plasmid, they don't have the resistance to tetracycline. And they will die if you grow them in tetracycline. Do you understand? Like, as in like all of you, are you guys with me?
Yes.
Okay, I certainly hope so. Now
Yes.
let's, okay, thank you. It's so hard teaching online. I can't tell. I can see you guys. I can read the faces. Usually I'm pretty good when I look at someone and I will know if they're confused or not. Um, anyways, this is the exact same question. The only thing we're changing is using a different restriction enzyme. Okay, this time we're using the SMA1 to insert the gene. Okay. And then we're going to grow it in tetracycline. Do you think they will grow this time? Okay, so the SMA1 means it's going to go right here. Okay. And that's a problem because the gene insertion interrupted the TETR gene. Right? It's going into the middle of the tetracycline resistant gene. So even if the bacteria, in this case, even if they accepted the plasmid, it's not going to protect them from the tetracycline, right? Because we've destroyed the tetracycline resistant gene by inserting another gene in the middle of it, right? So that's going to be a problem. Right? All of it will die. Even those who have accepted the plasmid will die. All right? All right, let's do one more practice together. Okay, so let's say we insert with EST1. Okay, so just nutrients. Nutrients plus ampicillin. Nutrients plus tetracycline. Okay. With nutrients alone, growth, no growth, some growth. What do you think? Hopefully you know that one by now. I hope you think you know it's like lots of growth, right? Lots of growth. Okay, they're happy. You give them nutrients, grow, 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 grow. Okay, number two, what about nutrients with ampicillin? Okay, nutrients with ampicillin. Lots of growth, some growth, no growth.
Lots of growth.
Okay, and why is that?
Due to the ampicillin, it's not gonna
Due to the ampicillin, or do you mean due to the ampicillin resistant gene?
resistance. Yeah, the ampicillin resistance.
Okay, yeah. So that's a good start, but let's pay a closer. Let's take a closer look at this. The ampicillin resistant gene is right here, right? The blue, basically like the blue portion of the plasmid, right? If I am inserting the gene with PST1, that means the gene is gonna go right here in the middle of the ampicillin resistant gene, right? Do you understand that? Which means you interrupted the ampicillin resistant gene. Even if they take up the plasmid, is not going to protect them, right? So this would be no growth. Aziza, is that okay? Like, does that make sense?
Yeah, I understand that.
Okay, great, thank you. Now, let's take a look at the last one. What about nutrients with tetracycline? Nutrients with tetracycline. Do you think they're gonna grow? Lots of growth, some growth, no growth.
No growth.
No growth? And why not?
Um, the tetracycline is what is it?
Is the antibiotic? Yeah.
Antibiotic as well, right? And
But
you're
then
inserting the PST, one
yeah, so the gene goes here. But the
as well.
tetracycline gene is over here, right? On the other side, right? You see?
Yeah.
Right. So the tetracycline gene is not interrupted in this case. Right. So that's why if they accepted the plasmid, they will grow. But not everyone will grow. Only the one that has the plasmid will grow. So this is some growth. Okay. So like basically what we have to do is we have to understand whether the insertion is going to interrupt the antibiotic resistant gene. If it interrupts it, then it no longer works, right? So what restriction enzyme is going to interrupt the tetracycline resistant gene? Look at the plasmid map. Which restriction enzyme is going to interrupt this tetracycline gene to make it like not functional anymore?
Is it that BAMHI?
That's right. That's the one. That's the BAMH, right? So like if you insert the gene using BAMH, then ampicillin will be some growth, but tetracycline will be no growth. Okay, let me just write that down for you. Insert with BAM.
So it's basically the opposite.
Yes, but do you understand why though?
Yes.
Yeah, it's opposite because you are interrupting the other
Yeah.
resistant gene, right? So if you interrupt it, they will no longer have protection against it. And that's why they will die in the presence of that antibiotic, okay? So this one is some growth, and then this is no growth. All right? And when I say insert with PST1, I just want to reiterate what that means, okay? To insert with PST1 means I will treat my plasmid and my DNA with the same restriction enzyme, in that case, PST1. And they will create compatible sticky ends. So the gene is going to go into the plasmid. That's what it means. Okay, that's the end of one huge concept. Took us like 2 classes to go through it. Any questions before we move on to the next big concept, which is DNA fingerprinting?
So what's the name of this concept again?
Recombinant DNA. Right? So this is an example of recombinant DNA helping to produce insulin, for example. Okay, recombinant DNA is taking DNA from different sources and putting it together. In this case, DNA from the bacteria, the plasmid, and DNA from humans, the insulin gene. Put them together, feed it to the bacteria, and then they will do transcription, translation of the insulin gene to make insulin. All right. Okay, let's take a look at the PowerPoints. So the next thing we're going to look at is DNA fingerprinting. You probably have heard of that before, right? DNA fingerprinting is useful for identifying, like matching a sample to a person. You can also use DNA fingerprinting to determine, who's related to who. But to understand that, I got to teach you a few other concepts. Okay. First one here is a polymorphism. This is not the first time I talked about this word. in a simpler sense, polymorphism is any differences in DNA sequence that can be detected between individuals. I told you before, any two humans are 99.9% identical on the DNA level. So the 0.1%, right, all those differences that exist, those can be detected with different methods. We call those polymorphisms. And these differences can happen in both coding and non-coding regions. Coding regions are your exons, right? You might remember. And then the non-coding region, they are the introns. We don't fully understand why there are a bunch of non-coding stuff that are in our DNA. Okay, maybe we just don't know what they do yet. Right? But for now, we view them mostly as fillers. Okay, like people call them junk DNA. They don't really have any apparent function. So one of these polymorphisms, variations that could exist in the non-coding region is what we call short tandem repeats. Okay, short as in like a few nucleotides long. Okay, like 3 nucleotides, 4 nucleotides. That's short. Tandem is one after another. Okay? If you walk in tandems, that means you're like lining up one behind the other, right? And repeats is, repeating itself. So over here we have CAG, 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 CAG. So it's short because it's just 3 nucleotides, and then it's tandem, one after the other, and it's keep on repeating itself, right? So we can write it like this: CAG 4. Right? The CAG is repeated 4 times. Now, sometimes these short tandem repeats are associated with diseases. An example of that is Huntington disease. Right? For those of you who don't know, Huntington disease is basically going to be a progressive neurological disorder that causes uncontrolled movement as well as loss of cognitive ability. And if you're thinking like uncontrolled movement, isn't that like Parkinson, right? Or like if you think about loss of cognitive function, and you're like, isn't that like Alzheimer's? They are all kind of under the same umbrella of disorder. They are all considered as neurodegenerative disorders, right? It's something that affects the brain. Okay, and so what they found is that there is a gene called the HTT gene, and within this gene there is a short tandem repeat of CAG. And scientists have found that in normal individuals, as in like people who don't have Huntington disease, the CAG is repeated anywhere between 10 times to 35 times. Okay, so if I say CAG 10, then it's like CAG, 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 repeated 10 times. That's what it means. Okay, so up to 35 times, that seems to be the normal case. However, there is an upper limit, a threshold beyond which people starts to get Huntington disease. 36 or more, okay, and as many as 120 times. That is what they found in people with Huntington disease. Okay, so this is like a dominant condition actually. So if you think about a person who has like big H, small h, and then they marry someone who's like small h, this person is gonna have Huntington. If you remember your genetics, 50% of the kids would have a chance of getting Huntington disease, right? And the symptoms don't really show up until, early 40s, by which most people would have already have kids, right? So that puts some people in a very scary situation where, their parents, one of their parents have Huntington, and then, you know, they know they have a 50% chance. And it's actually something that you can test for. You can do a genetic test, and it will tell you whether or not you are going to have Huntington. And you know, some people don't want to find out, right? Because, you know, they want to live life to the fullest. And, you know, if they test it and they're positive, it feels like, you know, they cannot enjoy life anymore, I suppose. Because, you know, potentially you'd be very sad about it, right? But then on the other hand, some people want to know, right? They want to find out. They want to make preparation. They want to, you know, not have kids, maybe not start a family, knowing that you could pass it on to the next generation. So there's some, you know, tricky stuff going on there. So that is Huntington disease with the short tandem repeats. But in terms of biotechnology, we can do what's called STR analysis as a way to distinguish, you know, one person from another. I mean, you can sequence the whole genome of the person, right? That then we would know. But then that's impractical because it takes too long, and, you know, there's time and money constraint, and sometimes your DNA sample is incomplete, right? So you wouldn't be able to sequence the whole thing anyways. So we use these STR analysis. So for person one, he might have the tandem repeats of AGAT repeated 12 times. But for another person, it might only be repeated 5 times. And these things will be found in the same chromosome, right? Chromosome 4 and chromosome 4, for example. And then the GATA both repeated 15 times, and then both of these would, you know, one of them repeats 30 times and the other 20 times. So if we keep on comparing them, then we will see that the short tandem repeats actually creates a unique sequence for everybody. Now, how do we exactly do this? Well, to understand this, I have to teach you another technique. This is called gel electrophoresis. Okay, gel electrophoresis. So let me just take some notes for you on the PowerPoint. I mean, on the OneNote here. Okay, and then I'll show you a video that shows you how this actually works. So this is called gel electrophoresis, which is a method used to separate DNA based on length. So we have a chamber here. It's just like a plastic container, nothing special about it. And you're gonna fill it up with buffer. Okay, buffer solution. The purpose of the buffer solution is to conduct electric current. Okay, conduct electric current. Okay, so you will hook it up to a power supply that sends a current to the black wire. Okay, and then it's gonna go through the solution, and then it's gonna go to the positive terminal. Okay. DNA backbone is negative because of the PO4, -3. Right? Sugar phosphate backbone. Right? I hope you remember I taught you that. So the phosphate is -3, which you learned in chemistry class, which means the current, as in the electric current, will push the DNA from negative to positive electron. Okay, so if you load your DNA here, I'll tell you about the loading in a bit. Load your DNA sample here. The current is going to be, oh, this is too thick. Hold on. The current is going to push the DNA towards the positive side. Now let's take a closer look at the gel. What is the gel? The gel is it's like Jello, okay, like really hard Jello, if you would. And how do you make Jello? I assume you are familiar with the process. Jello comes with packets of these powders, right? So it's the same thing. The gel comes in powder, and you dissolve the powder in some liquid, right? And then you microwave it, right, to heat it up to help dissolve the process. The more powder you use, the denser gel is going to be. And then you pour it into a mold, and then after it cools down, it becomes solid, just like the Jello. After you pour it into the cup or mold, right, you leave it in the fridge, it cools down, it becomes solid. So the gel is really smooth if you look at it, okay? But if you zoom in, like if you look at the microscopic structure, the gel is actually made up of these strands of fibers. Okay, they're called like agarose fibers, and it forms this mesh. Okay, the gel is a network of fibers. It's really smooth when you touch it, like the Jello. But then, like if you zoom in microscopically, it's gonna look like this. So let's say this is the negative side, and then this is the positive side, and you're gonna have the current. You're gonna have the current going through it. Okay, current. And then I'm gonna have my DNA. I'm gonna have some really long DNA, and then I'm gonna have some really short DNA in my sample. Because the shorter DNA is, well, it's short, so it's gonna be able to weave through these mishmash no problem. Okay, as the current is pushing through it, it's gonna be able to get through. On the other hand, this one is really long. So the current is pushing it, but like, it's not really going really far. It's getting tangled up in this network, and it's not going to be able to migrate as far down. So that's the theory. That's the principle behind the gel. Okay. It's like an obstacle course, right? Like one time I took my kids to like an indoor playground, and then they have this structure. I don't know if you guys ever seen this before, but I never saw it until that day. And it's like this gigantic cylinder in the center, like it's about 2-story high. And then they have several levels in there, several levels here. So like you climb up to the top, and then these are like they have tapes basically, right? Tape here in each of the level. And then I guess the point is for you to get in on the top, and then you can kind of make the way at the bottom. But you got to get through all these tapes, right? So my daughter was like, I want to race you to the bottom. I was like, okay, sure, let's do that, right? But because you're so much smaller than I am, like physically smaller, she's able to get through the tapes really quickly, right? Me, on the other hand, like I get stuck, right? And it's not an easy thing for me to do. So same thing here, because the DNA is longer, it would get stuck here and there. It's not going to be able to migrate to the end as quickly. But I eventually made it to the bottom. I eventually made it for the bottom. It's not that I can't, it's just that it's going to take me more time. So when you apply the current, you don't want to keep the current on forever. If you keep the current on for too long, eventually the long one will also make it all the way to the end. Okay, and that defeats the purpose, right, of the experiment. You want to apply the current a little bit, and then you stop, right? So given that time frame, the small one is going to be able to go further. The long one is not going to be able to go as far. So let's take a look at what this will look like. Okay, so here we have a gel. Okay, so here's the gel, and then we have some wells at the top. Okay. The wells is where you load your DNA sample. Okay, load DNA sample into the well. This is negative, this is positive, and then when you plug it in, the current will go this way. So at the end of it, after you apply the current, you will push your DNA down. Okay, the longer DNA will not be able to, like, this is the lane, right? This is the lane. You're not going to be able to go like sideways like that, okay? You got to stay in your lane. The DNA has to stay in the lane. So at the end, you will have like the longer DNA here and then the shorter DNA. Okay, so that's long DNA, that's short DNA. Okay. I know the line looks the same. The line actually does look the same, but what's important is, like, if you zoom in, if you zoom in to look at this here, right, they would all be like short DNA. Like short DNA. If you zoom in here, they would all be like long DNA. Okay, long DNA. Okay. Let me show you a video. I gotta pause the recording here, otherwise it's not gonna let me post it. So, you load the gel, as we saw in the video, and this is the chamber, and then we finish now. But at this point, we cannot see the DNA. We never was able to see the DNA because it's invisible, right, to a naked eye. And so what happens is we have to stain it with this thing called ethidium bromide. Okay, long story short, this thing binds to the DNA and it makes them fluoresce, makes them glow when you shine a UV light at it. Okay, you know, in like those crime shows, crime TV shows, right? Maybe you have seen it. They trying to see if there's blood in the crime scene. They turn off the lights and then they shine light, like UV light, and then the whole room lights up or something like that, right? You might have seen that before. This is kind of like that. Okay, except it's not blood that we're detecting. We're detecting DNA, right? So like you see there's DNA here, you see DNA there, right? So this is longer. This is negative, this is positive. Sorry, this is shorter, I mean shorter, because they are further down the gel, right? That's how you can tell that they are shorter. They were able to migrate further. But exactly how long, right? Like how many base pairs are we talking about? Well, it turns out you can also buy these things called leather from it's called DNA leather, and you can buy it from you know, again, the pharmatech company, biotech company, and the leather is just a bunch of DNA fragments of known length. Okay, so you know the length of each of these lines in the leather, so it serves as a reference for you to compare your sample to. So like you see a line here, and you're like, okay, let's compare it to the leather. Okay, it's about 268. That's all I mean. Okay, so you know, let's take a break first. When we come back, 5 minutes, we're gonna put all these together, and hopefully it's gonna make sense. All right, see you in a bit. Let's keep going. So next note is STR analysis. Okay, this is gonna take up some space, so make sure you you know, don't cram it into like a tiny spot on your note. Uh, I think I have to turn off my camera here. I had some problem this morning as well. Like when I did this part, like it was just like really slow with the camera on. I'm not sure why that is the case. It's okay. You don't have to see me. So here for the STR analysis, let's say we have two people. Okay. Say we have two people. Okay. Bob. And over here, we're gonna have Jane. Well, let's take a look at Bob's DNA. And we're going to look at chromosome 3. And let's say on chromosome, and you need colors for this, okay? 3 colors, otherwise the note will not make sense. So let's say on chromosome 3, we have an STR here, okay? And that is like, ATG. Sorry, that won't work. ATC repeated 10 times. Okay, and then we're going to have another chromosome that we're going to look at. And this one is going to be AC repeated 5 times. Okay, that would be on like chromosome, I don't know, 10 or something. And then we're going to do one more. We're going to look at another chromosome, chromosome 18. And then there's going to be another short tandem repeats. And this one is TTC repeated 20 times. And for Jane, we are going to compare, we're going to look at the exact same chromosome on Jane. Exact same chromosome on Jane. Like that. And the tandem repeats, the STR is going to be at the same spot. The only difference is the number of repeats might be different. So let's say this is repeated 20 times instead of 10. And let's say this AC over here is repeated also 5 times. And then over here, we have a TTC repeated, let me think of a good number, 5 times. 5 times. Okay. Because we know where these tandem repeats are located in the DNA, we can design primers to amplify these short tandem repeats. Okay, so you would get a sample, you would get a DNA sample from Bob. And this is easy. You can take it from like a cheek swab, right? Like swap the inside of the mouth, and then you get some cells there from the cheek, and then you would add the primers to it and run a PCR. So those primers will allow you to only copy these tandem repeats. So you will amplify this section. You will have like a billion copy of these ATC repeated 10 times, a billion copies of this repeated 5 times. Everything else is it's not going to be amplified. Only these short tandem repeats. Okay, so we're gonna have Bob's sample here. This is from Bob, and it's gonna be amplified by. So STRs are amplified by PCR. So within this tube, within this tube, you're gonna have a bunch of green DNA, so to speak. Of course, they're not colored in real life. Okay, this is solely for the purpose of like visualization. You have some blue ones and you have some red ones. Okay. Same thing for Jane. We're gonna also amplify her DNA using the same set of primers. And then it's gonna be like this. All right. Let's move it here. Any question before we move on to the next part? Okay. Now let's do some calculations here. How many base pair is this? If it's ATC repeated 10 times, how long is the total DNA strand? Give me a number. You know, ATC, ATC, ATC. You repeat it 10 times. How long is the whole thing? Okay. 30, right? 30 base pair. 10 times 3. That's 30. For Jane is 3 times 20, right? Which is 60. AC5, that's 10 base pair. Over here, this is TTC repeated 20 times, which is 60, right? And then over here, we have 15. Any questions so far? I know I just asked you that like a minute ago, but I need to double check. Okay, well,
I have a question.
yes, please.
Why are others multiplied by 3 and this AC is multiplied by 2?
Because there are 3 things that are repeating here, right? If you look at the ATC, there are 3 letters that are repeating.
Okay.
Yeah, and here are only 2 letters that are repeating, right?
Okay.
Actually, I am sorry. I want to make a small change here. I'm just gonna change this to 15. Okay, so 3 times 15 is 45. Let's do that. It's gonna be less confusing if we do that. And so I'll give you a moment to copy all this. Let me know when you're good to go. So is it possible unrelated people can have same STR? Yes. Like the blue one here, right? They are the same. Keep that point in mind. I will come back to it in a minute. That's a good question. All right, I hope we are all together. You got the numbers. Now we're gonna run this on a gel. We're gonna load the sample on a gel, the gel that I was telling you about. Okay, so we're gonna have a few lanes here, a few wells. Okay, we're gonna load it. You can choose any lane. Okay, I'm gonna put Bob's here in lane number 2, and then we're gonna let's put James right next to him. Okay, negative, positive. Okay, I really need you to work with me here. Okay, I'm gonna load the sample. I'm gonna run the DNA, and then I'm gonna stain it with the ECT bromide. So I want to ask you, right? There are three fragments in Bob's sample: the 30, the 10, and the 45. Which one is gonna be all the way at the bottom? The red, the blue, or the green? Which one is gonna be all the way at the bottom?
The green?
Why is the green all the way at the bottom? Let's see.
Because it's heavier.
Because it's what?
It's heavier.
Oh, it's heavier. Oh, okay. Now we don't know if it's heavier or not. This
Okay,
is
think of the number.
the number. Okay, so what does the number represent? Is the number representing the mass, like how heavy it is, or what exactly is the number? We want to clarify that. The number tells you how long it is, right? It's 45 base pair long, meaning there are 45 letters in it. 45, TTC, 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 right? And this is only 10 base pair long. This is 30 base pair long. Right? So in the gel, which gets to go further down the lane? The long one or the short one?
The short one.
Exactly, right? So the blue is the shortest. That's why it's going to go all the way down to here. Okay? This is the shortest one. Shortest. Goes the furthest. This is the 10 BP. Which one is closest to the well? The red or the green?
The red.
The green?
The green, because the green is longer, right? 45 is longer than 30, right? So that's why we're gonna have it like closer to the well. That is the 45, right? That's why I asked you to use colors, right? And then the 30 is gonna be somewhere in between the green and the blue, but probably closer to the green, right? Because 30 is closer to 45 than it is to 10. So this is what Bob samples is gonna look like. He's gonna have 3 lines at those locations. Now let's take a look at Jane. Jane also has the 10 base pair, the blue one. So she will have an exact same band at this location as Bob. But what is her longest one? Is it red or green? Hopefully you can tell.
Red.
That is the red, right? So the red is going to be closer to the top. But it's not going to be like here, because like if you compare it to Bob, that's like 45, right? And now we have 60. 60 is going to be even closer because it's even longer than the 45, right? So it's going to be higher up. And then with the green one is 15. So that's pretty small. Right? It's probably going to be like all the way down here, very close to the blue. So by using this technique, by analyzing the STRs at 3 separate locations, in this case, we can tell Bob and Jane apart. So, if there's a crime scene and then, I find like a DNA sample at some crime scene, whatever, and then it has like 3 lines, and I know it was Jane's sample at the crime scene and not Bob's, right? So that's basically how DNA fingerprinting works. Now, Lemieux asks, is it possible for unrelated people to have same STR? Yes, which is why we don't just do 3 STR for analysis in real life. We have to do many of them. So let me just go back to this picture for you, right? Like at the beginning when I was telling you about DNA fingerprinting, each of these lines represent a particular STR. Okay? So in this picture, these are all samples. Sorry, these are all like maybe suspects. I don't know what the context is, but like maybe these are different people. Okay. Suspect 1, suspect 2, 3, 4, 5, 6, 7, 8. Okay. And then we have a sample to compare with sample. So you look at the sample and we have all these bands. And look, this band is same as that band. This band is same as this band. Like the banding pattern is identical to person number 8. This is his sample. You know what I mean? That's his sample, basically. All right. Everyone else is going to be clear because the banding pattern is different, right? Do you understand? That's why we have to look at the whole thing, all the STRs that we know, not just 3, because just by chance, if 2 people have the same ethnic background, they might share similar STR. Siblings, they will share more STRs than the general public, right? Okay. So essentially, you can think of this as like a unique barcode for yourself, right? No one will have the same barcode, not even your family, right? Your barcode would be a combination of moms and dads. Okay? Your mom and your dad, right? Depending what was inherited. Remember, mom only passed on half of hers and dad passed on half of his, right? So you will have a combination of those bands together. Okay. I hope this makes sense. Anything you want to ask? Okay, so this is just to show you again, like if we're looking at 2 STR here, you know, this person has a really long one. That's why it's all the way at the bottom, at the top, and then very short one all the way at the bottom. Right? And you can just compare them, right? And you will see that these are from like 3 different individuals. All right, we're gonna move on. Because of time, I'm just gonna quickly go through these one with you. The plasmid is treated with equal R1 and BAMH1 when the digested plasmid is separated using gel electrophoresis. How many bands are you going to see? So like, you know, take a rubber band if you need to see this physically. The rubber band is like the plasmid. And then, you know, we're cutting it with equal R1. So that's 1 cut right here. And then we're cutting it again, BAMH, that's 2 cuts. Right? So if you take a rubber band, you cut it 2 times, you actually end up having 2 bands. 1 band here, another band here, 2 bands. Okay? It's different from cutting like linear DNA, right? If the DNA is like straight, it's not a loop, right? And you cut it 2 times, then you actually have 3 bands, right? So you gotta be careful. Like, are we cutting linear or are we cutting circular, right? There are 2 DNA samples with STR polymorphism. Sample A contains CAG10, the other contains CAG15. Which sample will be further down the gel when you separate it? Well, 10 is shorter than 15, right? So it's gonna be further down. So sample A. This one is 30 base pair. This one is 45 base pair. Okay, but you didn't need to calculate a total to figure out. Like 10 is shorter than 15. That's enough, right? Provided that the number of repeats are identical, right? Sorry, provided that the number of nucleotides in the repeats are identical. Like they're both CAG, so they're both 3, right? Okay. This one is not super important. I'm just gonna spend, you know, a minute explaining it. This is another kind of polymorphism. It's called RFLP. So polymorphism just means variation, right? Differences in the DNA. And this is differences in the fragment lengths created by restriction enzymes. So we're gonna treat the DNA with restriction enzymes, and then we're gonna see how many fragments are produced. So just backtrack a little bit. Back in lecture 7, I was telling you that there are silent mutations that distinguish you and me, right? So like you might have like a CCGG in your genome, and then just because, you know, somewhere in my ancestral line, someone had a neutral mutation, and instead of like CCGG, they switch one of the C's, and then it becomes like AT, right? So like you would be CCGG, I would be like CAGG. And it's just it's not like it's good or bad. It's just the way it is. It's just a silent mutation, and you know we all have our own silent mutations that we inherit from previous generations. Okay. So if we have a DNA sample here from like a crime scene or whatever, and then we have a suspect's DNA here, how do we know they match or not? Sure, we can do the STR analysis, but this is just another way to do it. So you can treat the sample with a restriction enzyme. Okay, and let's say this restriction enzyme cuts CCGG. CCGG is a palindromic sequence, right? So there are two CCGGs in this case. So you'll cut it once and you'll cut it twice. And then I will have 1, 2, 3 fragments. And when you lower it on the gel, you will see the same thing. Now, in the suspect's DNA, okay, if this happens to be a different person in this case, his DNA contains a silent mutation that causes the CC to become a CA. Okay, because the mutation happens to be in the recognition site, when you treat this with the same restriction enzymes, you will only cut it once here. Right? The recognition site is gone. Because of mutation. Right? So it will cut it once, and then you will have 1 fragment and 2 fragments. Okay? This fragment here is going to correspond to that one, which is actually the same fragment as the other person. But then, the top one is really, really big, right? Like this one is really big. That's why it's all the way at the top. In fact, if you do the math, if you add up these two, you will get back to that length. Okay, if you get confused by that, the key thing to remember here is this. When you treat a DNA sample with restriction enzymes, it will cut it differently for different people, because different people would have these mutations that would cause the restriction recognition site to disappear. Okay, that's all there is. So if it's cutting differently, your DNA, my DNA, and when we run it on gel, the fragments would be different in lengths, and so we get a different banding pattern. That's essentially what it is. Now I want to spend the last, 15 minutes talking about the next concept, which is gene therapy. Okay, gene therapy. Gene therapy, the hard stuff is done, by the way. Okay, there's no more crazy notes to take. This part you can just sit back and enjoy. Okay, it's fairly simple. So gene therapy involves introducing genetic material into a person's cell. Okay, presumably the person has some kind of illness, right? So that's why they say the patient's cell. And you can introduce it to their somatic cells, which is anything except for the germ cells. Germ cells are cells that will become sperm and egg, right? So if we don't touch them, and we introduce the gene to any of the cell body cells, like the liver cells, muscle cells, bone marrow, and whatnot, right? That's called somatic gene therapy, and that's only going to affect you. It's only going to affect the person who's receiving the gene therapy. It's not going to affect the future generation, because none of that change is going to end up in the sperm or egg, not going to get passed on to the next generation. Germline therapy, on the other hand, involves modification of the germ cells, the cells that will actually become the sperm and the egg. In that case, it will not affect the person who's receiving it, but his offspring, right? His children. So germline gene therapy is a no-no when it comes to ethical concerns. Okay, people think this is unethical because we don't have the right to affect an unborn person's genetics, right? Like what if it turns out to be bad, right? Then the person, the kid is gonna have to grow up suffering the consequences for something that they have no control over, right? The counter argument is this: what if we can enhance the future generation by introducing certain beneficial changes? Right? And this is true. You're gonna learn this next semester. Some people have a genetic mutation that protects them from HIV, so they don't get AIDS. It's impossible for them to get AIDS. Okay, and we actually know where the mutation occurs. So people are saying, hey, why don't we introduce this mutation to the germ cells? Okay, and then the future generation will be born without having to worry about HIV, right? That sounds like a legitimate argument, right? But then the counter argument for that is you don't know what actually is going to happen if you make this permanent change. Okay, like what if there are some unintended consequences that we are not understanding? And you won't know until the baby is born and grow up to be a person, right? Like how do you guarantee there's nothing's going to be wrong, right? So there's a lot of ethical debate, right? Basically, somatic gene therapy, green lights. Okay, people have done it. There are successful cases. Germline therapy, not so much because of ethical reasons. All right, why do we do gene therapy? There are 3 goals in general. Number 1, replace a non-functional gene. Number 2, suppressing an overly active gene. And number 3, modify the clinical course of the condition. I'm going to explain each one to you. Number 1 is probably the most obvious goal. You have a disease that has a mutation that causes a loss of function mutation. Okay, the protein is no longer working, so it makes sense to introduce the functioning gene, and then the cell will function again. But that would only be applicable if the condition is recessive. Okay, so let's say someone has sickle cell anemia. Okay, so they are like, little s. These mutations cause the hemoglobin to be misfolded, and it's not functioning. So I introduce a big S, the normal copy. As soon as the big S is introduced, it will override these recessive ones, and then you start making normal hemoglobin, and then it's fixed, right? This wouldn't work if it is a dominant disease. So let's say in some other disease, okay, whatever it is, okay? Let's say Huntington, okay, just for the sake of argument. Big H and small h. Okay, I introduce the normal copy. The small h is the normal copy. But it doesn't matter. Even if you introduce a small h, the big H, the one that's causing the problem, is still there, and it's dominant, which means it will override any recessive normal copies, right? So this kind of function, this kind of gene therapy is not suitable for diseases that are dominant. Okay, so things like sickle cell anemia, beta thalassemia. This is a nonsense mutation that causes your hemoglobin to be missing a chunk. Hemophilia is a bleeding disorder. For your blood to clot, there are like a bunch of clotting factors that needs to be working. Okay, and hemophilia, depending on which form, you're missing some of those factors. So when you bleed, you don't stop, and you have to go to the hospital to stop the bleeding in some cases. My grandmother has hemophilia, and even from like a nosebleed, because in the winter it's really dry with the heat and everything. If she has a nosebleed, like sometimes it would be bleeding for like hours, and then we have to take her to emergency to stop the bleeding. So that's quite scary. But these are caused by non-functional genes. So if we introduce the missing gene, we introduce the gene that will make the clotting factor that's missing, then we fix the problem. But it would only work if the effects of the disease are reversible, like the blood clotting situation. As soon as your blood starts to clot, you correct the conditions, right? In some cases, the disease already have done some lasting damage in the body. So fixing it like sometime later on in life is not going to undo all the damages that has already occurred. It's going to prevent further damage, but whatever harm was done, we cannot reverse that. Okay. Number 2, we can suppress a gene that is unwanted. You remember learning from me the oncogene, right? Oncogene is like that green light that wouldn't turn on. It just keeps on providing growth signal for the cancer to grow. So here we can use gene therapy to introduce a blocking gene. If the blocking gene is taken up by the cancer cell, it will shut down the oncogene mutation, and then it will go back to normal, right? The tumor is not going to grow further, and then you can use surgery and chemotherapy to remove whatever is there. Okay, so that's suppressing an overly active gene. And then number three is modify the clinical course of a condition. Right. So the aim here is to introduce. There are two kinds. I'll just show you the picture. It's easier. There are two kinds of way we can modify the clinical course. And again, this is ideal for things like cancer. Number one, we can introduce a suicide gene. Right? The suicide gene, once taken up by the cancer cell, is going to create toxins that would then kill the cancer cell. Of course, here we have to be careful. We don't want to accidentally deliver the suicide gene into healthy cells, right? Because then now you start killing healthy cells as well. So, being able to have a target delivery, that's the key here. Alternatively, instead of introducing a suicide gene, we can introduce a marker gene. Okay, so the marker gene, basically, once the cancer cell takes it out, they're gonna make these targets on their cell membrane. Right? So these are like targets. They're like, glowing lights telling the immune system, "Hey, I'm here, right? Come attack me." That's basically what it is, and that's what we want. We want the challenge for the immune system to identify the cancer cell is because the cancer cells originates from your own cells, so they blend in really well, right? They look very similar to your own cells. Okay, that's why your immune system is not able to identify them. So by introducing this marker gene, you make them different. You make them stand out from your own cells. And then the immune cells will see them quickly, and then it would just, kill them. And then, in the best case scenario, you will get rid of the tumor, the cancer cells, theoretically. So those are the three objectives of gene therapy. One last slide here. Gene therapy can be done ex vivo. Ex vivo means we take the cells from the patient, and then we're going to do some modifications in the lab, and then we put them back in. Okay? We re-implant the gene back into it. And this is usually done with bone marrow cells. Okay, because bone marrow, they contain like white cells and stuff, so we can modify them, we can educate them. We're like, hey, look, this is what the cancer cell looks like. I'm going to put you back into the body now. Go find them and kill them. Okay, that's ex vivo. You're doing it outside of the body. In vivo would be introducing the gene into the body. So that's a little bit more complicated because when it's outside of the body, you have full control of what's going on in the dish, right? Okay, and you know exactly what you're putting it into. But then when it comes to in vivo, you're relying on a vector to deliver the genetic material. The vector is usually a virus, like a virus shell. But once it goes into the body, we don't really know where it is gonna go, right? Okay, we're not 100% sure if it's gonna go to the target that was intended. And so there's a lot of risk associated with it. A lot of challenges that needs to be overcome. So that is that. I think that's everything you need to know for the quiz. We will still have a class on Tuesday. So like maybe don't rush into doing the quiz 6 a.m. on Tuesday. Maybe come to class first. And then we can finish the lecture and talk more about it. And then you can do it. But next week we're gonna basically do lecture 10. And then that will be it. Okay, we're gonna have one review class for the exam for the final test, and then the course will be done. All right, that's it for today. Thank you for coming. As always, enjoy the rest of your week. Bye for now.
You too, sir.